Hi everyone, it's me, and we're back again with our series of the circulatory system, and this will probably be the last lecture. I think it'll be a shorter one than the others. We were talking last time about the electrical signals that are sent through the heart that allows for the heart to contract, for the atria to contract first, and then the ventricles. And so we finished off with this picture, and we actually had it showing as an animation showing the different sequence of electrical events in the heart, okay, with the SA node, which is the pacemaker, going down to the AV node, and then down to the ventricles, and then boom, okay, and both sides are doing it at the same time. That's what we finished off, and we also talked about if you don't have your pacemaker cells working properly, you can be implanted with a pacemaker, okay, which is a relatively minor surgical procedure. A lot of people have pacemakers, and sometimes those pacemakers not only can regulate the heartbeat, but it also can, if it has a defibrillator in it, it can shock the heart, and for a heart that is out of control, get it back into rhythm. Okay, and then we finished off talking about what a heart attack is. And a heart attack is literally death of some of the cardiac muscle, and it all depends on which part of the heart and what level of damage, whether the person is going to survive or not. And so a heart attack is due to a clog or a partial obstruction or a full-on obstruction in a coronary artery. So the arteries that we have looked at so far, there are actually arteries that also go to the heart to supply the heart with nutrients and with oxygen. And if you block a coronary artery, then it's that portion, that tissue is not going to get any oxygen and it'll eventually die. And that's what a heart attack is. And so what causes these occlusions or these blockages? Well, it could be a clot that could be due to damage in a blood vessel, maybe from somewhere else that dislodged and the clot came and lodged into the coronary artery. Or more likely, it could be what we call clogging of the artery through fatty deposits and cholesterol over time from having a diet or having a lifestyle that contributes to that. And so here's a picture of a normal artery. And you can see this part here is the smooth muscle. This area in here, the white, is called the lumen, and that's where the blood flows. And so you can see it's nice and wide in this normal artery. And then this is a picture of an artery that has a huge plaque, a huge fatty deposit that is almost completely clogging this artery so that there's only this part left where the blood can go through. And so that will impede blood flow, could cause a heart attack. If it happens in a cerebral artery in your brain, it can cause a stroke. And here is a picture, and this is kind of gross, kind of comparing a clogged sewer to a clogged artery. They look very similar. And in fact, they have a lot in common. This fatty deposits here, very similar to the junk you find in a clogged sewer that will back it up. Okay. So I want to leave that area right now and move on to the structure and function of those blood vessels that we talked about. We didn't spend a whole lot of time on them. We spend most of our time on the heart. So now I want to look at the blood vessels that come from the heart and go to the heart, and then the ones that are in between the arteries and the veins. And so here's a nice picture from your textbook showing the relationship between the different blood vessels. We have the big arteries. Remember, arteries are large vessels, and so are veins. Okay, but arteries are carrying blood away from the heart. Veins are carrying blood towards the heart. And this would be an artery that is carrying blood away from the heart after it's gotten oxygen from the lungs, and that's why it's red here. Not all arteries are going to be carrying oxygen, okay? In fact, the pulmonary artery, if you remember, is the most oxygen poor vessel in the body, okay? It's on its way from the heart to the lungs. Okay, but in this case, we're showing an artery that actually is oxygenated and now is carrying that blood to branch points off the artery smaller arterioles. And arteriole really means baby arteries or little arteries. And then each of the arterioles is going to branch into what we call capillary beds, which are even thinner. And in fact, the capillary beds are only one cell layer thick. And then those capillary beds are going to merge into small veins called venules, which will merge into veins. And so then the biggest artery would be the aorta, and that would be even wider than this major artery, and it would come down here, and the biggest vein would be the vena cava. Okay, so that's basically the order that we've gone over before of blood flow. But what I want to look now at is the structure of these things. As you can see, both arteries and veins have connective tissue and smooth muscle, and then an area that the blood flows through, which we call the lumen. There's also a layer of epithelium that goes up against the lumen, and that epithelium is like skin cells. It's the same type of cells that you have in your skin, and also the same type of cells that line your digestive tract. Okay, you see the same thing with the veins. You have a layer of connective tissue, a layer of smooth muscle, a layer of epithelium, and then that lumen. You also were going to have these valves in the veins, especially in the extremities. We talked about those. Now, you also will see that in arterioles and venules, you're going to have this same type of um, connective tissue and smooth muscle and stuff like that. But I want to make the point between the artery and the vein. And we mentioned before that the walls of the artery are thicker walled than the ones in the veins. And that's because they have to withstand a greater pressure because the blood is coming straight from the heart to the arteries, where the pressure of the blood by the time it gets to veins is much lower, and so they don't have to be as thick walled. And so what you see here is you can see the smooth muscle is thicker, the connective tissue is thicker than in the veins. But it also means that the opening for arteries is actually going to be smaller than the veins. Veins have actually a wider diameter for their blood to go through than in the arteries. And this artery, this smaller diameter of the artery, actually maintains that blood pressure. So it keeps that blood pressure really high. Okay. We're going to talk a little bit about these arterioles in a little while. But first, I want to talk more about the capillaries. Now, these capillaries, as I said, were one cell layer thick, and they're very, very thin. Okay. And that makes them beautifully suited for exchange. And so we're going to actually get exchange of nutrients, exchange of wastes, exchange of gases, oxygen and carbon dioxide only at the level of the capillaries. You get no exchange from the blood vessels to the tissues in the arteries or the veins or the arterioles or the venules. They're too thick. Okay, you can't diffuse across those walls. But once you get to the capillaries, they are only one cell layer thick, and things can diffuse right across them. Okay, and so here's a picture of a capillary. Capillary, as you can see, they're just one cell layer thick between the lumen where the blood goes through and the actual layer of the capillary. And so what will happen is if you look at what's going on with oxygen, for example, oxygen is being carried in red blood cells on that protein called hemoglobin. And what will happen is once it gets down to the tissues and you want to drop off that oxygen, the oxygen will jump off the hemoglobin, diffuse across the red blood cell, diffuse across the capillaries into this fluid here, and then diffuse into the tissues. Okay, and diffusion is really, really slow compared to pumping blood really fast. But by the time you get to this place, you're right next to the tissues. So it only has to diffuse a really small distance to get into your cells from the capillaries. And so that's where diffusion does occur. Okay. Diffusion, if we would allow diffusion to occur from the lungs all the way down to the tissues and just kind of diffuse down, as I mentioned before, it would take about 5 years. So we limit the diffusion to when you get to the place you want the oxygen to go. okay? And same thing with wastes, and same thing with carbon dioxide and nutrients. So that's the only place where you're going to get exchange. Okay. Now, um, you might ask, well, how fast does this go? Is blood flowing really, really fast through here? Is blood flowing really slow? How does it all work? And so in order to do that, in order to look at which places you get blood going fastest versus slowest, let's look back here. So the question I'm having now is, does blood flow fastest through the arteries and then slower to the arterioles and then even slower to the capillaries, or vice versa? Does it go slowest through the arteries and then fastest through the arterioles and then even fastest through these thin capillaries? Okay, you could say the same thing for the other side. That's the question I want to ask. What is the speed? Okay, and is it suited for what we want? And so in order to look at how fast blood flows through these different vessels relatively to each other, we have to look at something called the law of continuity. And what the law of continuity, it's a law, so it can't be broken. It's a law that states that a given volume of fluid, so if you have a certain volume of fluid and you're sending it through a pipe or a blood vessel, that set volume of fluid will travel faster through narrower segments of the blood vessel than through wider segments. So fluid will flow, a given amount of fluid will flow faster through narrower segments than wider segments. Okay, so how does this work? Well, let me show you. Let's say you have a blood vessel, or you have a pipe, or whatever, or a hose would be even better. And you have a narrow segment, and you start a certain amount of fluid that's coming through here. Okay, you're not reducing the amount, you're not increasing the amount, you just have a set amount of fluid, and it has to go through. Okay, it will go through this narrow segment faster, and then will slow down over the wide segment and then speed up again. So it'll kind of go like this. It'll go at a fast rate through here, really fast rate through here, and then go slow as it goes through here, and then boom, speed up again. So fast, slow, fast, like that. And the way to remember that is to just remind yourself of what you've probably done before when washing a car with a hose. So you turn on the faucet with the hose attached, and you've got that set, set diameter of the hose, and it's not coming out very fast. So in order to make it come out even faster, what do? You squeeze down on the hose. And if you squeeze down on that hose, you are reducing the diameter of the hose, making it narrower, and boom, the water comes out even faster, and you can wash your car or spray your friend. Okay, that's what we used to do. So we'd let it be going normal, and all like that, and it sprays your friend. That's because of the law of continuity. It's going at a certain rate through this um, wide segment in the case of the hose, and then you clamp down on it and it goes really fast. And then if you unclamp down it, it'll slow down again. And then you clamp down on it, it'll go really fast. Well, that's the law of continuity in action. And that's very important for blood flow through the circulatory system. So if you have a certain amount of blood, blood's flowing as the heart is contracting, it's going through, and it will go through narrower vessels faster than wider diameter vessels. Okay, that's what you would think. And so now when we look at the law of continuity with respect to the three vessels we looked up at here, if we compare arteries versus arterioles versus capillaries, which one will it go through the fastest? According to the law of continuity, we should look at this and figure it out. So now we have an artery with a certain diameter, and if we compare that to an arteriole, the arteries are, each artery is wider than each arteriole, and each arteriole is wider than each capillary. So capillaries are the thinnest, then arterioles are a little bit wider, and then arteries are the widest. Okay, So based on that, you would think, based on the law of continuity, that it would go through the capillaries fastest, then the arterioles, and then the arteries. And if you thought that, if you thought it should go through capillaries, then arterioles, then arteries, in that order, going from fastest to slowest, you'd actually be wrong. Why is that? By the law of continuity, it should go through a single capillary the fastest, a single arteriole the fastest, and a single artery, I mean, slower, and then a single artery the slowest. But in fact, it goes through arteries the fastest, and then arterioles a little slower, and then those capillaries even slower. So what's up with that? Sorry about my cat. Come here, Chicklet. What's up with that? Well, you got to think about what the circulatory system really looks like. It doesn't have just one artery becoming one arteriole, becoming one capillary. You actually have branches. And so it looks more like this. If you look at the biggest artery in the body, which is the aorta, that is the widest of the arteries. And then you do get a smaller diameter for the major arteries, but you branch off into multiple ones of them that branch off the aorta. And then each of the arteries has multiple arterioles branching off. And then each arteriole has a big, huge capillary bed. This would be a capillary bed branching off each arteriole. And it's the total surface area, the whole diameter of the surface area that you need to take into account in determining which one is the widest versus the most narrow. And so because you don't have just one aorta, one artery, one capillary, and one arterial and one capillary, and then the same on the other side, it doesn't go slowest, faster, faster, even faster, and like that, because it's the total surface area that's important. So one aorta is going to branch into a larger surface area of arteries, which is going to branch into a larger surface area of multiple arterioles, which is where it's going to go even slower. And then the capillary beds are all over the place, and there's tons of surface area there. So it will slow down even more at the capillaries. And that's what they're designed to do. You want it to go really slow once they get to those capillary beds, because that will allow you to have time to exchange. So things go really slow through the capillaries. And then once exchange has occurred, you want to get that blood back up to the heart as soon as possible so it can dump off the CO2 and pick up oxygen again. Okay? And so that's where, once it gets to the venules, okay, those overall are going to be a smaller diameter than the big capillary bed, so it speeds up. Getting to the veins overall, because there's less veins than venules, smaller diameter overall, speeds up even more. And then the veins merge into the vena cava, which gets it even faster. And that helps it to go up, even against gravity, that speed. Okay, so that, even though it sounds like it would be the reverse, you have to take into account that total surface area. So exchange occurs the slowest through the capillary beds. And actually it makes sense. Remember, we said that we're not going to have any exchange at all in the arteries, arterioles, venules, or veins, or the aorta or the vena cava. And because you're not going to have any exchange, you want stuff to go as really fast as possible. And that's what the whole purpose of the circulatory system is, to try to get things from point A to point B really, really fast. And then once you finally get to B, then you slow down and allow for diffusion to occur. So I like to think of the circulatory system as sort of like the Orange County. And you think of the major arteries as being like the freeways, like the 405 or the 5 or the 22 or something like that. And then off those freeways are multiple off ramps, like, from the 405, we might have Magnolia or Bolsa or Beach Boulevard. Okay. And so things will slow down a little bit there. And then as you leave the off ramps and go on to, say, Beach Boulevard, then you can start going, as you get into the capillaries, into the side streets. And you get to your side street and you dump off your oxygen. Okay. And then when you want to go back to work or whatever, you have to go and speed up again. Okay. So that's the way I like to think of the circulatory system. Okay. Um, we have one more thing to talk about with respect to these blood vessels, and that is that the blood vessels can also be regulated in terms of their width and how narrow they are at any given time or how wide they are at any given time, which will help regulate blood flow to a particular organ or tissue. Okay, and so see the smooth muscle that we have here, particularly here, a little bit here as well, but you also have smooth muscle in the arterioles, and it is the smooth muscle that can constrict, that is, it contract, or relax, and that will tend to close the opening more if it contracts, or if it relax, open up the opening even more. Okay, and so that can help regulate blood flow to a certain tissue. And so I'm gonna talk only about the most important vessels in this regulation, and that is the arterioles. Yes, you can do it at the level of the artery and somewhat in the vein, but the arterioles is where you really are going to regulate blood flow to certain places in your body. And so let's have an example. The example would be you're just kind of hanging out and you are just had a nice meal, you're digesting and you're resting, and you're getting done with your digestion and you're getting up and you're kind of at a resting state, and all of a sudden a serial killer comes after you. Okay, this is morbid or whatever. They're not going to kill you because you're going to be smart. What are you going to do? You're going to get the hell out of there. You're going to get into what we call fight or flight mode, and you're going to amp up your everything, and those skeletal muscles of yours in your arms and legs are going to need to contract fast so you can run. Okay, so what do you want to do? Do you want to continue sending blood to your digestive system in massive amounts so you can digest and hang out? No, you're going to want to switch things up. You're going to want to divert that blood from your digestive system and now send it to your blood vessels that are going to go to your skeletal muscles so you can get the hell out of there. Okay, that's what you're going to want to do, and there's a way to do that, and it's basically at the level of these arterioles for the most part. Okay, so that's what we're going to focus on. And so those arterioles, as I mentioned, have a lining of smooth muscle lining the vessels, and that smooth muscle, if you're just hanging out, not doing anything, not digesting, not running, they have a certain layer of tone. The smooth muscle is always a little bit contracted. And so when it has that little, what we call arteriolar tone, that's when the vessel is at rest and it has a certain diameter of opening and a certain amount of blood flow will go through to the tissues. So every tissue now is getting pretty much the same amount. Now, if you want to suddenly change that tone so that you can get less blood to one place and more blood to the other, well, you would do these two things in different areas. If you wanted to get less blood flowing to your digestive system, then the arterioles lining those digestive capillaries, you would want to constrict. And so you would contract that smooth muscle more, and that is called vasoconstriction. Vessel constriction. It's called vasoconstriction, as seen right here. Vasoconstriction means the whole vessel diameter decreases because the smooth muscle lining it is contracting. So that means less blood is able to go through there, less blood to the digestive system. Now that blood has to go somewhere, and so you will vasodilate somewhere else. Well, you vasodilate by relaxing that smooth muscle in the arterioles that lead to your skeletal muscles. So you can divert that blood very quickly. And that's how we do it. And there's lots of different ways that you can do this. Different chemicals will do it. Some gases actually will do it. That different hormones, different drugs will do the constriction or the relaxation, vasodilation, vasoconstriction. but I'm going to look at one that is natural in your body that plays a big role in doing this vasoconstriction and vasodilation, depending on the tissue it's looking at. And that is a hormone. Let me make this a little bit bigger. Sorry. It's a hormone called epinephrine. I mentioned epinephrine before. Epinephrine is also known as adrenaline, but from now on we're going to call it epinephrine. Now, epinephrine is your fight or flight hormone. It's released from one of your organs called the adrenal medulla right into your bloodstream when you are under any kind of stress, major anxiety, fear, even happy stress, okay? But it's designed to get you ready to run away, okay? And so it's going to do a ton of things to your body systems. It's going to increase your heart rate. It's, as we talked about before, it's going to increase your stroke volume too, so it increases your cardiac output. It's going to do a lot of other things as well. It's going to increase your blood pressure. It's going to help open up your airways. But what it's going to do to these arterioles is it's going to either constrict or dilate that smooth muscle, depending on which arteriole. So take a moment to think about if we were to look at the arterioles leading to the digestive system or the arterioles leading to the skeletal muscles like of your legs and arms. What would epinephrine do in each one of those? Remember, the goal is to ramp up your body and get ready, okay? And so epinephrine actually has different roles, does different functions on that smooth muscle depending on where the smooth muscle is, in which arteriole beds, arterioles leading to capillary beds. So epinephrine is going to actually act like a vasoconstrictor, okay, for the arterioles for the digestive system and any other system that you don't need when you are running, okay, or when you're scared. So it's a vasoconstrictor for all of those arterioles, for all those systems that you don't need when you are in fight or flight mode, okay? On the other hand, epinephrine is going to be a vasodilator for the skeletal muscle. Okay, so it's not vasodilating skeletal muscle. It's vasodilating the arterioles that lead to the skeletal muscle because your skeletal muscle needs a blood supply as well. Okay, and so it's going to allow for more blood flow going to the skeletal muscles and less blood flow going to the digestive system, which is exactly what you want when you want to get out of there. Okay, and so that's the one that I want you to know. Epinephrine is usually, it actually, in most systems, it's going to be, in fact, a vasoconstrictor. But when it is working on the skeletal muscle, it actually vasodilates, which is what you expect. Okay? So, would not want to vasodilate the digestive system when you're running around, running away from somebody. Okay, so we're going to end it there. The last thing I want to talk about this is that you're going to see me talk about, when I talk about epinephrine, I'm also going to talk about the sister molecule that's very similar to epinephrine called norepinephrine, which we just abbreviate NE, norepinephrine. Okay, and so norepinephrine and epinephrine are very similar hormones, but they're also neurotransmitters. So these two molecules have multiple roles in the body, and they're mainly roles of fight or flight. Okay, and so they can be, if epinephrine is acting like a hormone, it'll go through the bloodstream and act on certain tissues through the bloodstream, and norepinephrine can do that as well. If it's acting as a neurotransmitter, it also can be released from nerve endings, sort of like acetylcholine, okay? And then it'll act on certain muscles or glands or something like that. So it has both roles. It can be a hormone, it can be a neurotransmitter. It's got a million roles in the body, but we're going to mainly focus on its role as a chemical hormone, meaning that it's released into the bloodstream and works on certain target tissues. Okay. And when we talk about epinephrine, we're also encompassing norepinephrine, but we're just going to call it epinephrine. So I'm trying to make it easier. From now on, I'm just going to say epinephrine when I could be meaning both. And I'm just going to call it a hormone, but be aware it can sometimes be a neurotransmitter. Okay. And later on, we're going to talk about what hormones do. They are chemicals that are released from certain glands, certain organs, and they are released right into the bloodstream. And then they go around the bloodstream looking for target cells. And those target cells will have receptors on the surface or inside them. Okay. And those are the only cells that are going to act. In this case, the smooth muscle would have an epinephrine receptor. Okay. Now, just to bring back this hormone thing, which again we'll talk about later, we've already seen a couple of hormones in this class. Try to think about which hormones we've talked about. We've actually talked about, that I know of, at least two. One was insulin. Insulin was a hormone, and remember we talked about what insulin does. It's released from the pancreas when you eat a meal of glucose. And then the pancreas releases insulin directly into the bloodstream, like all hormones. And then the insulin runs around the bloodstream looking for target cells that have their receptor. Okay? And then it binds to the receptor and has an action on that cell. The other one we saw, actually within this series, was erythropoietin, EPO. EPO is also a hormone. It's released from the kidneys. It then travels through the bloodstream looking for a target. The target is the bone marrow. There are EPO receptors in the bone marrow. It binds to the receptor and gets some action. That is, makes the bone marrow make more red blood cells. And so we're going to, as I said, my last lecture is going to be all about these hormones, but I want to kind of give you a head start on what a hormone is and how some of the hormones work. And epinephrine is a big one because it works on so many different systems in a fight or flight mode. Okay. Now, that's it for that way of regulating blood flow. Okay, you can use epinephrine to either vasoconstrict or vasodilate, depending on the tissue, and it's always released when you're in fight or flight mode. But there's another way that you can regulate, in general, blood flow, not through the arterioles, not through the arteries, but at the level of the capillary beds. Okay, and so remember this picture right here. Now we're talking about these capillaries. We just talked about how you can regulate the diameter of the arterioles. Well, how can you regulate the capillaries? You can't really regulate the diameter of the capillaries. They're only really tiny in the first place. And in fact, if you were to squish down on the capillaries, you wouldn't be able to get red blood cells through them. Okay, so you can't regulate the diameter of the capillaries, but what you can do is you can regulate whether blood actually gets into a capillary bed at all. And so the other way that you can regulate blood flow to a certain tissue is with this little smooth muscle, layers of rings of smooth muscle that line the entrance to a capillary bed, and they're called precapillary sphincters. A sphincter is a ring of muscle. So again, smooth muscle is going to play a role here. Okay, smooth muscle was playing a role with the arterioles. Okay, now smooth muscle is playing a role in the capillaries, but not on the inside of the capillary, but around the outside and at the very entrance. Okay, and so right now, these precapillary sphincters are relaxed. And so as blood comes through the arteriole, the blood can go to all of the capillary beds. And so, like, let's say this is the capillary beds that line your skeletal muscle, let's just say. Or no, let's say that this is your digestive system. So these are the capillary beds that go into your digestive system. Okay, so Blood is coming through here and everything's open up. Okay. Now, if you wanted to slow down blood here, you can slow it down by vasoconstricting. But if you want to slow it down even more and not let it go through these capillaries and let it go straight through to the venule, then you can clamp down on these precapillary sphincters as well. Okay, and so right now they're open, blood goes through all the capillary beds, but you can also close it, and if they clamp down, then the capillary beds don't get the blood. Instead, it goes straight through what they call a thoroughfare channel, right back to the venule and right back to the heart. So you completely bypass that capillary bed by clamping down on those precapillary smooth muscle rings. So there's two ways that you can regulate um, how well blood flows through a certain area. One we just mentioned with the arterioles, you can slow it down or you can speed it up, or not let very much blood go through if you vasoconstrict, or let a lot of blood go through if you vasodilate. But then you can, at the next level, also regulate by clamping down on those sphincters, and then blood will go right back. So you might have an arteriole that you have vasodilated, and it leads to a bunch of capillary beds. Okay. And so now the blood's coming through in a large amount because the arteriole is vasodilated, and you want the blood to go to this capillary bed up here, and you want the blood to go to this capillary bed over here, but you don't want it to go to this capillary bed. So you keep the sphincters open on this one, you keep the sphincters open over here, but you close down those sphincters here, and so this capillary bed is bypassed. And so that's another way that you can regulate blood flow through your body. Okay. The last thing we're going to talk about, and this is going to be the last part of the circulatory system that will be on the third exam, is going to be blood pressure. Okay. And so blood pressure, it's actually a measure of the pressure that the blood exerts against the walls of arteries. And here we're looking at the systemic arteries. We're not looking at the pulmonary arteries. We're looking at the ones that are branching off into your body. Now, we also don't look at the head one because you really can't take the carotid artery and do a blood pressure cuff around that, although you could measure it, although the person would probably die. So what we do instead is we look at to measure blood pressure, we look at the arteries that are in the extremities, like the brachial artery, which is your arm, or you can also look at your femoral artery, which is your big artery of your leg. You can take blood pressure there as well. So it's basically measuring the pressure of that the blood exerts against the walls of those arteries. Okay, and so when we do blood pressure, we see two numbers. For example, 120/70. Okay, and so what do these numbers mean? What do they represent? Well, the upper number, which is always going to be higher, represents, we call that the systolic pressure. Okay, the upper number. And remember, in systole, you are contracting the heart, you are contracting the ventricles. Okay, systole is ventricular contraction or heart contraction. And so, what that systolic pressure, that number represents, is the pressure that the blood exerts against the walls of your arm artery when the heart is contracting, when the ventricle is contracting. So, it's going to be a high number compared to the lower number, which is going to be during ventricular relaxation. So, that's called the diastolic pressure. And so there's still going to be pressure of the blood against the walls of the artery when the heart is relaxed, okay? But it's going to be less than when the heart is contracting. So those are the two numbers. Blood pressure against the walls of the arteries when the heart is contracting. The upper number, that's the systolic, and then the lower number is the diastolic pressure, which represents the pressure that's still there against the artery walls when the heart is relaxed. Okay, so how do we measure blood pressure? Well, the best way to measure blood pressure is actually with a cuff around your arm to occlude or close down the brachial artery. You don't want to use those wrist blood pressure cuffs. You can use them, but they're not that accurate. Okay. In fact, that's the ones that we were going to be doing in the lab. But so that's why we have to take everything with a grain of salt. And you also want to take your blood pressure a couple of times in a row and average the numbers if they are within closer ranges. If they're way off, then you'd have to take it a third time. Okay. So what you do is, let's say the blood pressure is going to be 120 over 70. How is that measured? Okay, how does a nurse or a doctor figure out those numbers? Well, here's that brachial artery. You put the cuff around that artery, and then you inflate it with air. And what that air does is it puts pressure on the artery. So you have to bring the pressure on mercury, which you can read with the blood pressure um, mercury reading, pressure reading. I can never say the word sphygmont. I can't say that. Whatever. Um, you have to bring it over, obviously, over 120. You don't know it's going to be 120, so you usually bring the pressure and pump and pump and pump until it's up to about 160, 170. And that should be high enough to be above this person's systolic pressure. So it has to be high enough so it completely compresses that brachial artery and no blood is going through. That's why this is white. No blood's going through. Okay. And then you slowly let the air out. And as you slowly let the air out, you're reducing the pressure against that brachial artery. And eventually it'll get to a point where the brachial artery blood will start seeping through. And you'll actually audibly hear that with the stethoscope as lub dub, lub dub, lub dub, as the blood starts coming through because the pressure is less. Now, once it gets to the pressure of the systolic pressure, as you're reducing and reducing with the thing, once it gets to the point where it's starting to leak through, that means the pressure this way and the pressure this way is equal, and that is your systolic pressure. So that's your upper number. Okay. And so then you let it go and you slowly let out more and more air, and you'll hear the audible sounds and you'll hear the audible sounds until it gets to a point where the pressure is so low that that brachial artery is completely open now and there's no resistance at all to flow. And when that happens, which in this case would be at 70 millimeters of mercury, then the blood flows freely and you hear no more sounds. Okay. And so the systolic pressure is where you reduce the air flow pressure with the little blood pressure cuff, and it gets to a point where some of the blood leaks out, and you can hear it, lub dub, and when you can no longer hear it anymore, that's the diastolic pressure. Okay, and it takes a little bit of practice to do this, but it's kind of fun to do blood pressure that way. Okay. The other way with the little cuffs that we're going to do in the lab is kind of boring. You just watch it and it goes. Okay. Now, what does this all mean? Well, there are more people that have blood pressure in this country than know it, and it kills. It's called, in fact, the silent killer because most people that have high blood pressure, unless they go to a doctor and get it checked, and unless they have other obvious symptoms, they will never know it, and it can kill them. Okay, and the reason why it can kill them is because when your body is being exposed to that high pressure, it means your heart is working harder. Not only is your heart working harder and creating more pressure against the walls, but that pressure against the walls is starting to damage the arterial walls. And so the arterial walls start to get damaged and can get weaker and can get little injuries on them that can create little healing of the injuries, which can make a clot, and then the clot can dislodge and cause a heart attack or a stroke. And it can, as I said before, it will eventually damage your heart because your heart's working harder. It'll damage your blood vessels. So there's a lot of reasons why having high blood pressure is not a good thing. Now, there's causes of high blood pressure that are not always fully understood. Some of it's genetic, some of it's due to lifestyle. Having too much salt in your diet, that doesn't affect everyone, but for some people that are sensitive to it, too much salt in your diet as your kidneys try to bring the salt back in and they have to retain some of the salt, water goes along for the ride, and water increases in your blood circulatory system, increasing the volume, which increases your pressure. Um, Smoking increases blood pressure because it's a vasoconstrictor. Caffeine can increase blood pressure temporarily, so you should never take your blood pressure within 30 minutes of smoking or having caffeine. Being overweight increases. There's a lot of different ways to get blood pressure. Now, here is numbers that you will not have to know, but I bring this up because about 3 years ago, these numbers were different. Okay. In fact, you did not have high blood pressure at 130 over 80. 130 over 80 was not considered high blood pressure before. Before, it had to be 90 or higher for diastolic, and often was 140 over 90. They've brought the numbers down considerably, the American Heart Association, because they have more evidence now that individuals that have these numbers that are even lower have problems. Okay, and it's attributed to those numbers. And so it's a good thing to get your blood pressure checked regularly and make sure that you are in a good range. And so we're actually going to stop there. I know this was a long lecture. I thought it was going to be short. It's going to be a long lecture, but this is the end of the material for exam 3. Okay. And even though we're going to move on a little bit more with this PowerPoint in the next lecture, which is going to be the structure and function of blood, that will not be on this exam, that will be on the final. So this is the end of the exam material, okay? And it actually says that in the study guide, okay? So have yourselves a great day, and I will talk to you later, okay? Bye.